formed at each of 3 enzyme active sites. So a very unique way of creating ATP. Of course, this ATP that's created inside the matrix of the mitochondria is then going to be sent out to wherever the cell needs it. So, the point of this slide is to talk a little bit more of this mechanism, this rotation here. Right here, this is not pointing to the positive charge on arginine. I'll talk about that in a minute. But right here on this subunit, because over here, you see a proton is bound to that subunit. Remember there's 10 of these subunits that are identical? Well, there's an aspartate that is picking up that proton. So a proton is hopping through this first half channel, going on to this aspartate, and then going for a merry-go-round ride around and then out the upper half channel. So this arginine is in the A subunit, that's this thing here. And the arginine, remember, has a long R group, and it always has a positive charge. So I'd like you to learn that the arginine is doing 3 very important things. One thing is that the arginine here, with its positive charge, is typically, well, it's constantly moving back and forth. Its average position would be right here in between the 2 half channels. So that if there's a positive charge here, this is going to prevent the proton from cheating. It would be terrible if the proton could come up here and then out the other half channel. You see that? That would be bad, because now no work is being done, no useful work. So the positive charge on the side chain of that guanidino group of arginine is saying, proton, you may not come past me. I'm the guardian here between the 2 half channels. So that proton then has to hop on and rotate this thing, which turns the gamma subunit, which is what kicks off the ATP. Item 2 that the arginine does is that as this C subunit here that picked up the proton along with its brothers and sisters here, as this thing rotates around to the half channel that exits to the other side of the membrane, there's water and acid-base groups in this exit channel, just like there are for the entrance channel. But imagine you're a proton, and as you come close, part of you wants to jump off into the aqueous environment, and this positive charge here is an extra incentive for you as a proton to leave, because you're being repelled by the arginine. So the arginine is helping to kick off that proton. Thank you, arginine, because we want the proton to leave. The last thing that the arginine is doing is that once that proton leaves, that leaves behind a C minus, a C with a negative aspartate, so it's an aspartate minus. Well, in that short distance that it has to go from the exit channel to the inlet channel, that would be thermodynamically not allowed. Instead, the arginine is there to act as an escort as it goes across in that short little distance, because in that short distance then, there's no net charge. That's pretty cool. That allows the rotor to continue to spin. So thank you, Mr. Arginine. Arginine 210 is, the 210 part is not important. The point is it's a particular arginine that's sitting right there, just going back and forth. Okay, well now, here we've got the matrix making ATP. And once again, the protons are not going up the center here. They're going halfway up and around and then out. And the ATP is not being made at the end of this structure. It's being made at the 3 dimers. So we'll have to excuse Leninger for this error. But I want you to see that the ATP has to leave so that the cell can use the ATP elsewhere. But it only does so going through a transport protein, an integral protein, something that's buried in the membrane. And it only does so, it comes out if an ADP comes back in. So this adenine nucleotide translocase is very important. It's allowing the ATP to cross the membrane because ATP is quite charged. It can't go across the membrane unless there's a protein that provides a pathway. And that only happens if the ADP comes back in. But notice that there's something subtle going on here. How many phosphates are on the ATP? 3. Adenosine triphosphate. How many phosphates are on ADP? 2. So 3 phosphates are coming out, but only 2 are coming back in. Well, that would mean that within a few moments, the whole synthesis of ATP would stop because you can't make ATP from just ADP. You need the 3rd phosphate. So that's where this other transporter down here, phosphate translocase, is a symporter, which means 2 things have to come in at the same time. So inorganic phosphate, which is H2PO4-, along with a proton, is coming in, providing the missing phosphate. Cool? So this guy up here, this translocase, allows an ATP to come in if an ADP comes back in. So again, the pool size of adenosine diphosphate and ATP is tiny. So thank you. But then down here, the other translocator is keeping the phosphate pool intact so that you can make ATP. So what we see is that this one down here costs you a proton. So when you ask, how many protons does it take to make an ATP? On our previous diagram, we said it takes 3 protons to move the crooked finger 120 degrees, which releases an ATP, but it also costs you a proton to bring in the missing phosphate. We also see that this ATP coming out is electrostatically attracted to the charge difference here. So really the proton gradient is running both translocators, because the proton gradient is making it electrostatically attractive for ATP with its -4 charge to want to come out to this positive membrane. So the energy driving all of this is really the proton gradient. And of course, the proton gradient was driven by the energy in the form of electrons on NADH and FADH2. So that comes then, we come to this question, how many protons does it take? Well, as I just said, 3 to move that rotor, the gamma subunit, 120 degrees, plus the 1 to bring in the phosphate. So it's going to cost you 4 protons to make 1 ATP. Now NADH, if we go back to our previous, one of the slides that we had, I think we had 2 slides actually that summarize what happens when NADH binds to complex 1 in the electron transport chain. 10 protons get pumped across. So we can do the math and say 1 NADH creates 2.5 ATP. Now the half part shouldn't cause us any concern because this is a continuous process. The rotor is moving all the time, the electrons are going down all the time, so you're just making a fair amount of ATP. So I'll let you figure out how many ATP you get from one FADH2. Okay? Think about that. Anyway, you're getting a lot of ATP from the energy that originated in NADH. All right, well, now we're going to go to our last topic in our course, which is photosynthesis. And what we'll see here is photosynthesis has several things that we talked about previously, just with a slightly different variation in the chloroplast. So what do I mean by that? Well, we're going to see the Q cycle. We're going to see enzymes, not all of them, but many of them from glycolysis. We learned glycolysis a long time ago. We'll see the ATP synthase. And so many of the things that we've learned previously are applicable here. So today it's, hi, how are you? Photosynthesis. Look at some of the components that make up this interesting system in plants. And maybe get to photosystem 2. PS stands for photosystem. Roman numeral 2, there's 2 photosystems, PS1, PS2. I don't think we're going to get to the cytochrome B6F complex, but the cytochrome B6F complex is the chloroplast version of complex 3 in the mitochondria. Remember complex 3 was the thing that had you put your hands together to form 2 caverns, and it uses the Q cycle. This thing, called cytochrome B6F, has the same 2 caverns and the risky iron sulfur proteins going up and down. They're just called this. Okay, so we've got this overall scheme here going on. And it just says that organisms use energy, and here we are as humans over here, and I like the fact that we're not that much more important than the bacteria in this scheme of things. But as you know from biology, a lot of organisms derive their energy directly from photons of light. And so we need to focus here on these purple and green bacteria. Why, Dr. Bill? Because it looks like the Z scheme, and I'll bet you've learned the Z scheme a long time ago in biology, and we're going to inoculate you with that again. But it looks like the Z scheme in plants evolved from these 2 bacteria sometime in evolution. So that's the bigger picture. You're not memorizing the slide. The point is, I need to tell you about purple and green bacteria. So here we have a simplification of what's going on. Energy from the sun is being used to break water down to oxygen. There's a lot of water involved. Now it's shocking. And some of that energy is trapped in the form of NADPH and ATP. NADPH is the cousin of NADH. So you know the structure of NADH. This guy has a phosphate at a particular position. Well, a phosphate has a -2 charge. Enzymes are not confused by NADPH versus NADH. So typically, a rule of thumb is that NADPH is used to reduce things. So if you need to build a molecule like a sugar, and you need to reduce carbon dioxide, remember carbon dioxide is the very oxidized form of carbon, photosynthesis is going to go the reverse way to build sugar. And so NADPH should be thought of as a very high energy compound. ATP, you know, is a high energy compound. And it turns out that you need 6 NADPHs for every 9 ATP in the chloroplast to build sugar. And so all of this is done to take 3 carbon dioxide, which are 3 carbons, and make it into glyceraldehyde 3-phosphate, G3P. Where have you seen G3P before? Glycolysis. So 3 CO2s and a water are used to make a sugar. And it requires a lot of energy. So I need you to learn that the photosystems, remember there's 2 photosystems, PS1, PS2, they make this NADPH and ATP in this particular ratio, 3 to 2. 3 ATP for every 2 NADPHs. Not some other ratio, because I've told you many times that the pool sizes of things like ATP and NADPH are small. Hence, inside the chloroplast, so now we're inside a different organelle in the plants, in the chloroplast, there's a tiny pool size. The photosystems will make it in precisely this ratio, because in the stroma, the liquid that is around the thylakoids, that's where the enzymes of the Calvin cycle, named after a professor at UC Berkeley, who discovered this. The Calvin cycle uses these 2 compounds to build the sugars in precisely that ratio. There's no waste. It's very efficient. So the light systems are making it, and the Calvin cycle is consuming it. And it's just recycling these things. So long as the sun is shining, everything is good. Okay, so here is just a drawing of what would a chloroplast might look like if you cut it in half. You can see that there's a lot of membranes here. The thylakoids are these structures here with these membranes that have photosystems 1 and 2 in them. And out here is this liquid that is bathing all of these thylakoid membranes. This is called the stroma. The stroma is where the CO2, carbon dioxide, is chemically transformed by the Calvin cycle into sugars. So what I want to say is that this organelle has in this liquid some circular DNA, just like the mitochondria have some circular DNA. But just like with mitochondria, about 80% of the proteins necessary to build everything in the chloroplast are actually coded for by the nucleus. So most proteins have to be inserted. They come in from the outside. They're built in the cytosol, and they get translocated across membrane. This picture here looks like they're showing ribosomes building proteins out here. And so this organelle can build some of its own proteins, but not all of them. So there's really 3 compartments that we need to learn. One compartment is this liquid, an aqueous environment called the stroma. Another compartment is down inside these thylakoid membranes. So if you cut this membrane structure, there's liquid inside. This is called the lumen. I'll give you that spelling in a bit. And then you have the compartment being the interior of these membranes. So remember how coenzyme Q was a hydrophobic molecule that could carry electrons, does carry electrons and protons. It's imprisoned in the membrane of the mitochondria. Well, this chloroplast has a similar carrier that's hydrophobic and is zooming around inside its membrane. But it also has to have a carrier inside the lumen, that is a liquid part, there has to be a carrier. And there has to be a carrier out here in this liquid. So we're going to learn in photosynthesis that there's really 3 carriers, one in each compartment. Inside the lumen, which is liquid, inside the membrane, and also inside the stroma. So each one's got its own little carrier. Okay, well, we're not freaking out about this. If I drew these on the final, or drew one of them, you could say, oh, that must be this. These are compounds that have the ability to absorb light. Basically what that means is they have a lot of conjugated double bonds. Double bond, single bond, double bond, etc. So Leininger has put it into pink here, the conjugated ring system here. For chlorophyll A, that's this molecule here, and chlorophyll B has got some slight modifications. That's very tiny. So what I need you to know is that most chlorophylls have a magnesium here. This heme, so this structure here, it looks like a heme structure, but it's not. This porphyrin ring is what it really is. It doesn't work to absorb light unless there's a magnesium with a +2 charge there. So instead of iron with a +2, like in hemes, cytochromes and hemoglobin and myoglobin, this thing needs a magnesium. Sometimes these molecules of chlorophyll aren't chlorophyll if they don't have a magnesium, they do something else. Then you've got these compounds, beta-carotenes. They have lots of conjugated double bonds because they have a different structure here than this molecule up here. They absorb light at different wavelengths. Here, lutein has a slightly different structure than up here. It absorbs at different wavelengths than beta-carotene. So carotene, you can remember as carrots have a lot of this stuff in it. And sometimes students ask me, well, why do carrots, which grow on the ground, have all this carotene in it if it's not doing photosynthesis? And I don't have a good answer. The point is, when it's facing light, these compounds, all 3 compounds, absorb light at different wavelengths. And that's a good thing. So here is just a diagram showing the solid black line is indicating what the light coming from the sun typically looks like to some organism on the planet. And it's a dog-eat-dog world here. And so imagine if you're in a jungle, the leaves that are up at the top of the canopy can get whatever light they want, and the ones down at the floor of the jungle are saying, hey, I'm happy with whatever light those guys miss. And so you have pigments that basically have evolved to handle different parts of the spectrum so that the photons of light coming from the sun are not wasted. So something like chlorophyll A has a peak absorbance out here, but also one down here. But these other things with different structures absorb light differently. Well, all of this is terrific if these things feed their energy to a common destination. It doesn't really matter what light you absorb so long as you're sending energy to somewhere in the plant that can use this. So here's the part where we begin to lose me and everybody else, okay? We get into the quantum world. And what we talk about is an exciton transfer, which we don't truly understand. So it turns out that these photosystems have a reaction center called an RC. So imagine that Dr. Hilt is the reaction center, RC. Not shown in this diagram, but we'll see it somewhere else, either today or next time. Remember I told you that sometimes metals get really close to each other and they act as a binuclear site? We've seen that in complex 4, the mitochondria. 2 coppers get really close together and they act as one. Well, here in a reaction center, what makes me special, my hand here is the porphyrin ring of one chlorophyll, and my other hand is its sister. And they're positioned by proteins so close, they act as one. So I have a special, at the reaction center, I have a special pair of chlorophyll. And what happens is, everybody else here, all these colored things are supposed to represent pigments and proteins in the lipid bilayer. That's you guys. You're feeding me your energy. You absorb photons of light, and somehow you're sending that energy to me. And that's the black box that we don't fully understand. How does the energy from you, in the form of what we call an exciton, a quantum of energy, get from you to me? We don't fully understand it because of things like quantum tunneling and all sorts of bizarre stuff, okay? My best analogy is, you know how when we have clicker questions, I have this little antenna here, and you're clicking your clicker, and almost instantaneously, I get a message to my computer as to what you chose as an answer. So much faster than if it was a particle going randomly in the room. It would take forever for that particle to finally hit here. But as a wave, it can get here much more efficient. And so basically, it's a wave function because these things at such a tiny scale are waves. And they are very efficiently, you guys are sending your energy that the light is hitting to you like a radar dish. You're sending it to me at the center of the radar dish. And that energy allows me to pop an electron to a higher energy state. So if on the y-axis here we're talking free energy, G, I can take that energy and pop one electron up. But I can't do it again unless somewhere I get an electron to replace the one that I just popped up. Cool? So it's really an electrical circuit being powered by the energy of the sun. All right. So here, you're not memorizing this. We're going to take a look at these 2 bacteria that I mentioned previously, purple and green, to see how they work. And then we're going to say, it looks like the Z scheme of photosynthesis in plants fuse these 2 things together. Okay? So over here, purple bacteria have a reaction center. RC stands for reaction center. That's me with my special chlorophyll dimer pair. That I can pop an electron to a higher energy level, okay, if I'm in this bacterium. So excitons of light, photons of light have triggered the pigments in the photosystem of this bacterium, and they're sending excitons, which are quanta of energy, to me, and I can pop an electron up to a higher energy state. And so this pigment, P870, P stands for pigment. I absorb light. I have a color. They call me a pigment. 870 is just the wavelength in nanometers, which happens to be the maximum for my absorption spectrum. I plotted absorbance versus wavelength. So that's a way a human can denote what this thing is. On the y-axis here is the redox potential, E0 prime. Remember we said that's under 1 molar conditions except for protons. So pH70 gets excited to a higher energy state. And then it can lose an electron to a molecule called thiophytin. Well, thiophytin is a chlorophyll without a magnesium. So light uses a molecule in a different way by just getting rid of the magnesium. So the electron goes through the backbone of the proteins. All of these pigments are held by proteins in perfect orientation. Without the proteins, the circuit wouldn't work, just like in the mitochondria. That goes to a friend of yours. What's Q? Coenzyme Q. It's the same Q that we see in the mitochondria. Same molecule. Not a big surprise because we think bacteria were the beginnings of mitochondria. Then it goes to a cytochrome BC1 complex. Okay, that's a lot of verbiage for something that looks like the 2 chambers of complex 3 in the electron transport chain. So the electron comes into this and through a Q cycle sends an electron to cytochrome C2. That's analogous to cytochrome C in the electron transport chain, but it's not. This is not part of the electron transport chain. And then that electron goes back to the reaction center. I'm the guy who popped an electron. I can't do it again until that electron comes back to me. Then I can do it again. So why do all this? Because this complex here dumps off protons just like complex 3. Remember a QHQ comes in, 2 protons get dumped off on one side of the membrane. That's what this thing is doing. So it's creating a proton gradient. Why does it care? It wants to use that proton gradient to make ATP. So these guys, purple bacteria, can make a proton gradient from just using the energy of light. That's pretty cool. Whereas these characters over here can do one more thing. So green sulfur bacteria, their reaction center uses a different pigment. Not a shock then that its redox potential is not the same as this guy down here because it has a different structure. So excitons excite it. When it goes to an excited state, the electron can come down, just like over here, and go in a circle to make a proton gradient. Or the electron can go to a protein called ferredoxin. I'll give you the spelling in another slide. Ferredoxin's job is to carry one electron at a time. Can't carry a proton, just an electron. And that electron can go to an enzyme complex that reduces NAD to NADH and a proton, because whenever you write NADH, there's also a proton. So how many electrons does NAD carry? 2. So this would have to happen twice, because that's how you need 2 electrons to make this go to NADH. Now what they're leaving out is, here in these sulfur bacteria, if the electron which started here goes through this scenario to make NADH, then the electrons have to come back to this reaction center from somewhere. They're coming from sulfur, reduced sulfur. But they're not showing that in the diagram. Cool? So these guys can use light to generate a proton gradient. These guys can use light to either generate a proton gradient or make some reducing power in the form of NADH, not NADPH. Okay, so with that in mind, we see, this is the Z scheme that we find in plants, in cyanobacteria. And it sure looks like those two photosystems got brought together a long time ago in evolution. Some cell got lucky and got both bacteria. You are not memorizing this thing, but I need you to know who the players are. As it says in homework 3, I need you to know the concepts of photosynthesis. So, I need to talk about this diagram here. Here in plants, and we're going to focus on plants, P680 is a reaction center. It's in photosystem 2, PS2. So if I'm this guy here, I've got two chlorophylls that act as one. They can pop an electron to a higher energy state. When I do so, it shows light coming in here to excite it. That's possible, but usually the energy coming to me is from exciton, from all you complementary light-absorbing compounds like carotenoids and other things, just feeding me energy. And I'm so thankful. So now I'm popping my electron to a higher energy state. That can go to pheophytin, then it can go to something called plastoquinone A. PQA down here is plastoquinone. This molecule, as I'll show you in a future slide, looks a whole lot like coenzyme Q. But it's the plant version. When you see plasto, that means plant, or chloroplast version. Plastoquinone B is getting that electron from plastoquinone A, and it is mobile. And it will carry its electron to the cytosine B6F complex, which is the equivalent of complex 3, the thing with the two changes. Plastoquinone B, along with plastoquinone A, always carry electrons as H dot, just like coenzyme Q did. It can carry 1 or 2. So it can carry 2 electrons, and when it does, it always carries them as 2 H dots. Why does life use this version where one, PQA, doesn't move and the other one does? I have no idea. No one does. Okay, the electron that started here is going off, and it's going to eventually go over here to the other reaction center, P700, which is in the other photosystem, PS1. So, before we go through all of that, the point is that electron that originated here is never coming back. So there has to be a way then of, if I'm the dimer right here, I just popped an electron to a higher energy state, I'm going to have another electron replace it, otherwise I can't pop the next one. Well, that electron is coming from water. And there's something called the oxygen evolving complex, OEC. We don't completely know how it works, but this protein complex, its job is to split water into oxygen and electrons, and that electron goes to this, and so it can do it again. So what I'm going to show you here is P6